Welcome again to session number 4 out of 20, afternoon 2. I hope that the break was productive for everyone. I hope you're getting a lot of work done and gaining knowledge. Thanks, Rejoy. Everyone's so supportive here. It's a wonderful, wonderful environment. Gather room is still operating. I heard there might be some issues with the riot rooms. But as long as you're able to get in touch with somebody, if you're on a hiccup or having full-blown Docker breakdown, we'll be able to resolve the issues, and it will make everything go even smoother for the next 4 days. So again, don't give up. For now, we've got the final session for today. We're going to finish up with quality control of a genetic data set. We'll take a look at the summary statistics for major depression, and then we will run a instance of PRSice inside of our Docker and get to some polygenic scores. And using this same code and the same approach, you will be equipped to do the same type of thing for your own analyses, your own collaborative work, should you ever encounter genotype data. You'll know the right questions to ask. So go ahead and get right back into it. There we go. Okay. So we are right here where we left off. I will briefly explain some of the commands that we're going to enter for each round of Plink. More importantly, I'm just going to give an impression of how Plink syntax kind of works. You'll be able to reference the Plink website whenever you like and get key materials from there and more in-depth descriptions of how each call and each command and each option works in terms of what it excludes, what it includes, how it performs its quality control. The basic structure of every Plink command is the same. It begins with a call to Plink. Here in the bash format, this backward slash just indicates that the command is not over. It tells it to continue reading the line until there is no backslash at the end. So each one of these commands could be entered in sequence, separated by white space or just a space, but it's easier to see each individual command when it's written this way. So the first thing that Plink needs is a genetic data set. And you remember that the genetic data set consists of 3 separate files: the bed file, the BIM file, and the FAM file. And Plink knows this. It knows to look for those files. So all you need to give it is the prefix that is at the front of each of those files. Now, if you've named them differently for some reason, if you've just have a different name in front of your BIM file or your FAM file, you can also specify them individually. But the simplest call is to start with a B file command, which says, look for a full 3-file data set in Plink binary 1 format. So it'll look for the bed, the BIM, and the FAM. And all of our genotype files are in the Geno directory, and they all start with the prefix Geno R08 and 1000. It's always nice to include in your dataset prefix, and really anytime you're writing files out during a multi-step procedure, to keep track of what is in that file with something informative in the title. So rather than just say genotype data, you know that this is genotype data that has already been pruned so that the R, the imputation quality, is greater than 0.8 or 0.8, and that the number of subjects in that sample is 1000. In fact, this is a smaller subset of data from a larger data set that I had, just to help us kind of operate in the computational space more effectively and play to some individual machine's weaknesses in case there are any. Sample size of 1000 is a little more manageable. The following commands, I've outlined what they all do. Each one of these is kind of performing one of those initial steps that I talked about, both at the SNP and subject levels. You can read what each one of them is doing. We have the minor allele frequency threshold. Here, we're going to remove all SNPs from the data set that are at a frequency of 0.05. All allelic frequencies, anything less than that. Any allele that's found in less than 5% of our samples will be flagged. The SNPs that have that low allele frequency will be flagged in our analysis, and we'll be able to remove them downstream. This is the Hardy-Weinberg equilibrium flag. It will test the Hardy-Weinberg equilibrium for every single SNP, and if the p-value for that test is less than 1 × 10⁻⁶, which is quite significant p-value, if it's a strong deviation from the Hardy-Weinberg equilibrium, it will flag that SNP as well. We also have the Geno command, which will remove missingness of SNPs. So if there are certain SNPs across our sample that are missing data, that haven't been genotyped or imputed successfully for a number of subjects, it will remove it if it is actually anything, if it's missing anything more than 1%. And MIND does the same thing, but not for SNPs, for subjects. Any subjects that are missing anything greater than 1% of their data will be removed from the analysis. It flags them to be problematic. We're not expecting to have any samples with a fair amount of missing genotype data because these samples were imputed. They were imputed with high quality. The R is 0.08 or greater. And the final two commands have to do with output, really the final three. The two here, write SNP list and make just FAM, are to keep Plink from doing the thing that it will do automatically. If you just say, I'm applying these filters and I'm specifying out as a prefix to the files that it's going to spit out. In this case, we're going to be spitting out files that begin with geno_QC. We're telling it, instead of creating by default a whole new data set, right? Instead of taking the bed file and writing out a whole duplicate data set with a few of these filters applied, we only want it to write out a list of SNPs, right? We want to include just the SNPs that we can then put into a downstream commands, and the SNP list will be all of those SNPs that we want to include, all of the SNPs that pass the QC thresholds according to these filters that we've applied. And also, when we say make just FAM, we're saying, instead of, again, don't write a whole new large 2 GB genetic data set, just write the FAM file so that now we have, first off, the SNP list of SNPs that we want to include for our next analysis, and all of the subject information. Remember, the FAM file contains the family level, the subject level information. So this will apply these filters and then give us a list, a FAM file for all of the individuals that pass these filters, as well as a SNP list file that passes all of these filters as well. And this will save space. It saves a lot of space on your drive. It saves a lot of time in writing it. So we'll just copy this command, as simple as entering it, insert, run. And you'll see Plink will print the options that are in effect. It will read the amount of RAM that you have available on your machine or inside of your virtual environment and reserve a certain amount for its primary workspace. And it will get to work. The first thing it has to do is read the binary data set. That'll take a few seconds. But once it's read that binary file set into memory, it can process these types of flags and filters quite efficiently and quickly. So you see, immediately it's telling us some information, and all of this will be written to a log file, to the GenoQC log file, and we can examine it afterward. It's telling us that this genotype data set contains 9.3 million variants. So this is a complete, imputed, quite a large data set. It's telling us we have 1,000 people, and it's saying we have 0 males, 0 females, 1,000 ambiguous samples. And that's because we have zeros in that entire column of our FAM file. Remember, I'm storing phenotype information. Sex can be included in that. I'm storing that elsewhere. We've done sex checks, so don't worry about that. It won't flag any errors. It's just letting you know that it's writing also ambiguous sex IDs, writing out to this no sex file. Not important. We're aware of this. We know in our sample who is male and who is female. And let that run for a couple seconds. And we have a question. Copy, so instead of clicking the run chunk inside of our markdown, well, when I click the run chunk, it sticks it into the R console instead of into terminal. And I don't know how to get it to directly paste the chunk into terminal rather than into R console. So if you have a fix on the way to do that, then that would be very much appreciated. Typically, I mean, it works fine this way. In fact, using the terminal environment to run bash programs like Plink or like PRSice inside of this RStudio environment is handy in a lot of situations. So it's handy in this situation because we can run Plink and PRSice inside of a Docker image without having to install a second bash client or terminal program, and we can operate all of these things as it normally would. Yeah, it shouldn't run. I have a feeling that connections with Crowdcast seem to be changing the speed of running some of these programs. In testing, it's only taken a few seconds. So we have a few things. And Shu, error out of memory. The memory flag may be helpful. That's and a disappointing fix, a disappointing face. So that's fair. We can fix that. I'm curious how much memory it says was allocated, Anne, at the beginning of your run. I see. So interesting. Yeah, so that makes sense. So so Anne, you must have a, so I'm not sure if that can be remedied in Docker or if it's just your computer. Right, because 429 megabytes of RAM is not really enough to work with these types of genetic files, even slightly smaller, fairly smaller ones, although you may be able to change the amount of memory allocated to the Docker image. Daniel, is this for settings resources inside of RStudio for allocating more memory? Right, so this is the thing that I'm concerned. So typically it should be able to recognize, it should be able to, I mean, everything that I've run on here is through default settings, but I mean, it's only detected 8,000 megabytes of RAM. I have quite a bit more than that. It may be, it says command not found. So did you try running Plink with help before to verify that Plink is found inside of your Docker environment? So there are a couple of things. If there's command not found for Plink, the important things to check is, one, have you typed bash? Have you clicked bash so that we're running bash code? So you have to enter the bash command to have it recognize that. Second, if you can't, if it still is not recognizing Plink, if you just type Plink and press enter or Plink help and you don't get, and it says command not found, then you may have to try the workaround script that's back at the top of the R notebook, right here, this source line, source this if Plink not found. And if you run that and it still doesn't work, then the only thing I can think of is a syntax, a copy and paste error, something coming out of this. Because if you can copy and paste it, if you are on S. Yeah, it may just be a starting bash thing. So it's line 158, 159. Okay. Okay, good. So it sounds like, there you go. Yeah, give that a try, Radha. So we can just take a look at what that command did while we still kind of tweak Plink and make sure that everyone's getting Plink to go correctly. This explains those things to notice. The outputs that we specified are inside of Geno, so we can always look inside of the Geno directory to see what it gave us. You did that still, not happening still. So is it still, can you get in touch with somebody in the gather town to maybe send them the error message itself about not being found? Radha, so maybe if you're able to do that, then because beyond, okay. So let me know either way. Hopefully, you know, assuming that memory isn't going to be an issue for you, or we can get the Docker to allocate more memory. It all depends what you have on your laptop. So some, okay, Aaron will be there in GatherTown. So again, this kind of speaks to the fact that it is challenging to do this kind of research. So even for demonstration purposes, if you want to work with kind of some realistic data, computational requirements are there. There are certain computational requirements. You shouldn't need, and we tested the code on a number of different laptops. We're not using supercomputers, but there are certain limitations. And that's kind of the name of the game, unfortunately. But there are always ways, and again, because it's a Docker environment, this is the nice thing. If you're ever on another computer or you're another computing environment and you have Docker available to you, you could pull all of this code, do it the same way, and then use that system to try all of this that same way. So sometimes the computer will be a limiting factor, but to do multi-scale neuroinformatics, unfortunately, there are some resources that we need, and then sometimes those get in the way. There will be a bit of a workaround a bit further down, and we'll get there and I'll kind of explain how we can get around it. So the second set of QCs, well, I guess I'll quickly mention the files that it spat out. So you'll see, remember I said make all of the QC file outputs start with geno_QC. So it's given us a file called geno_QC SNP list. So that's the list of SNPs that passed quality control according to all of our filters. I can use cat, which just kind of prints and reads out any text file. I can take a look at the log file that was generated during that run. It's just a copy of what was printed out to the terminal. And remember, it's telling us the number of variants that were loaded from the BIM file, 9 million, the number of people, 0 people removed due to missing genotype data. That's good. That's what we want. Before the main variant filters, there were 1,000 founders and 0 non-founders present. This is related to family analysis. We're not doing any family analysis. We're not processing any of that. So everybody is a member of their own family. They're all technically founders, according to this definition. It's giving us a read of genotype rate, which is almost 100%. It's telling us that 928 variants were removed due to missing genotype data at Geno. So there were some people with missing genotype data. So you'll see things like this. I mean, these are numbers that you should get accustomed to seeing. Always we will have missing data in some place or point. When you get up to very, very large numbers, or if you have, 9 million, if you have most of your variants being removed due to missingness, it tells you that maybe something wrong happened in a previous step, a merging step, bringing chromosomes together, translating from different file formats like VCF or BCF into the Plink binary format file system. Each one of these kind of just gives you an idea of where you stand with the overall quality of the data set that you're working with. Here we have 13 variants that were removed due to Hardy-Weinberg test. That's fine. We also removed a lot due to minor allele frequencies. We removed over 3.5 million variants because they had too low minor allele frequency. That's also quite reasonable. In the end, high quality imputed variants with a reasonable minor allele frequency with no Hardy-Weinberg violation And all individuals that had a successful genotype missingness of less than 1%, we still have 5.5 million variants in 1,000 people. That's quite a good, solid data set. There's still just a couple more steps that we want to go through to ensure that we're not making any kind of cardinal errors related to our genotype file set. This one is heterozygosity. Remember, heterozygosity can indicate to us if there's been a sample contamination or high levels of inbreeding. And this command will do much of the same as what we saw before, except now we're reading the same original genotype file, but this time we have an extract command. So it's saying, don't worry about all 9 million variants. Just now look at the ones that are in our past QC SNP list. So we're extracting these 5.5 million variants that had the proper minor allele frequency, no missingness, etc. We're also going to tell it to keep, which refers to subjects and not SNPs. Keep only the individuals in this new GenoQC FAM file. Now, lucky for us, as I say below, no subjects were removed for missingness of data, right? It was high enough quality. So this GenoQC FAM file is actually identical to our original Geno R08 N1000 FAM file. We didn't remove any subjects, but in your general workflow, this is what you would do. In case 1, 2, 3, 10, 15 subjects were removed, now we're only going to look at those individuals. And then the het command will give us an idea of heterozygosity rates across our sample. We'll copy this and paste it in. Oh, well, you see, now I get an error, classic. Why did I get the error? Why didn't it run? Well, it's because in my example of showing you the files that were output, I changed directory into day 1 Geno. So I'm notified PWD, I'm no longer in day 1, and it's looking for a directory called Geno to put the output files, which doesn't exist inside Geno. So I have to CD back to day 1, PWD. Now we're inside of day 1, which is where the code is written to be designed for. Insert, run that again. So now it's going to go through much of the same procedure as it did before, except now it's not going to have to calculate many of the metrics that it did the first time. Now it's simply concerned with identifying heterozygosity rates. And it will spit that out in the same prefix of Geno_QC, but the file suffix, the name of the file will be .het. And we will use that file to then identify people who have outlying levels of heterozygosity, and then in the next step, remove them as well. It's a very iterative process performing this quality control. We start with one initial screen for variants and subjects, and then we go down the list just to make sure that we're ending up with the cleanest data set that we can. And we have, actually, the computer is still computing the previous commands. Well, it's great. It's great that there is variation in the way that it's running its commands. For our downstream, for our calculation using PRSice, we'll have the opportunity to choose between a very small kind of pruned data set that we'll generate to run a principal components analysis or the full. And when we get to that stage, I'll just point it out to everyone, we'll change the code, very tiny change, to use the pruned version. That should alleviate a lot of the memory demands that we have for this particular tutorial. So this bit here summarizes the measurements, the metrics that are used for determining heterozygosity across each subject. And now we can switch. This is one of the nice things about operating in console and in terminal. Having this RStudio interface allows us to switch between R and bash. We can now execute a block of R code. And the reason why we do this is because the het file that was just the het file was generated gives us measurements for each person. We want to now apply our own threshold, and we want to say, well, what do we believe constitutes an excess or not enough heterozygosity as expected? What do we believe lies outside of our distribution of heterozygosity? And that will be, in some cases, population specific. It's something that will be specific to our observed distribution of heterozygosity rates. So what this chunk of our code does is it reads in the GenoQC het folder using freed, which is a function from data table. which is a faster way of reading in data frames than just read table. Here it helps us locate the correct file in the correct directory, so that if your directory structure looks any different, you'll still be able to identify the correct file. And we're simply going to identify all of the subjects, the FIDs, the family IDs, and the IIDs, who have outlying measurements of heterozygosity. So that would be any F score. F score is a measurement that's calculated based on the distribution of heterozygous genotypes across the whole genome. And if anyone has an F score that is less than or equal to the mean plus 3 times the standard deviation, or greater than the mean of F minus 3 times the standard deviation, basically anybody who lies on the extremes of this distribution of heterozygosity, we will flag and write a file out that says, okay, these are the valid samples. Anybody who falls outside of this range of heterozygosity, we don't want to look at any further. And this fwrite command will write out a file called genoqc valid sample. It will write it inside of our geno folder, and then we can use that for our next plink commands so that we are, again, further whittling down our data to be the highest possible quality. So this should work as a chunk. You can execute it as an R markdown chunk, this little play command in the top right corner of the grayed out area. And I'm switched over to console here. I think it'll run it in console no matter where you are, if you're in terminal or console. But here you'll be able to see the code run. And it shows you a little green bar, command bar on the left-hand side to where the code is making progress. And once it's run the whole thing, you can see the R code is now in here, in your console. It's run without error. And now we can go and look for the file that we wrote out, this genoqc valid sample. And we wrote that out inside the geno directory. So we can use our ls command inside of terminal, ls the geno directory. And here we can see we have a file called genoqc.valid.sample. We'll use that for further subject pruning. Now, question, how many subjects were removed for outlying heterozygosity? Anybody out there want to have a dabble? Any ways in which we can take a look? Also, I can address a quick question while people are thinking about it. Is it common to remove variants less than a 5% minor allele frequency? It is fairly common to do that. Also 0.01, which would be a 1% minor allele frequency. Typically, it's a function of the sample size that you have and the quality of the imputation that you're working with. The more individuals that you're looking at and the type of genetic analysis that you're doing, the typically lower you can go in terms of minor allele frequency. That's primarily the limitation, just to avoid statistical pitfalls of variants where you have very few individuals with either rare, homozygous, or just heterozygous genotypes. So another question about using R markdown in the context for the workshop makes sense. In the real scenario, would the loss of resources be worth it instead of a bash script and an R markdown script for the plots? Same for Docker. So the resources, I mean, resource allocation in Docker should be flexible. I think when you perform Docker run, depending on the resources that your laptop has, you should be able to allocate as much RAM as you need. Assuming that you have enough. And the, I mean, resources shouldn't necessarily be lost, but you are right, Isabel, in the sense that the R markdown in this instance is certainly for the purposes of workshop. It's for the purposes of being able to take a look at the explanations alongside the code, and then in instances where the code cannot run correctly for you because of memory issues or because of any other kinds of errors, the knitted HTML document should contain the outputs of eventually what we want to get, so it allows for more participation. But you're right. Typically, if you are working in an environment like a Linux environment of a computing cluster, a remote server, which is most often how we do these types of analyses, especially on very big data sets, you'll be working in a Linux cluster. You'll be submitting jobs differently to a queue. You'll be interacting with these programs a little bit differently. It all has to do with the type of environment that you have and can kind of set up locally. Those who are on SciNet are doing kind of a bit of a tricky thing. They're still ending up with RStudio. The way that we want to do that is, I mean, the reason for this is for the workshop. So everyone's looking at the same thing. The windows look the same, just trying to keep it as uniform as possible. But you're certainly right. In many cases, it is not as efficient. So, and certainly for some cases where you're dealing with very large files and also dealing with very large outputs, to have your analysis embedded inside of an R Markdown object. For this case, it's actually quite, I think quite useful. So we have a couple of guesses. 10 people, a couple people of 10 people remove correct. Well, we can see that. I can just do a trick. I can cat this Geno valid sample file, GenoQC valid sample is inside the Geno directory, and I can pipe it. We don't have to go into this to get the word count with lines. So you'll see it's 991, which might lead you to believe, oops, probably where this 9 came from, because it's less than 1,000 and it's 9 less than 1,000. But the way that the file has been written, we head that file, you'll see that it has an extra row here for FID and IID. It has a header row. So it's not just a list of 991 subjects who are going to include, therefore meaning that we'd have 10 excluded, or sorry, 9 excluded. It also has one row for FID and IID. So in fact, there are only 999 subjects listed here, which means that is right. We've excluded 10 individuals due to outlying heterozygosity. What a shame. But that is par for the course, and that's fine. So from now on, we'll be concerned with a sample of 990 subjects in our genotype data. Next, relatedness. So before we get into calculating relatedness, and this is something that is also going to be useful for anyone who's having memory allocation issues, although it may actually be difficult to get this initial code to work, to actually read. If it's reading in the genotype file that's causing the issue, it won't necessarily help. And I can provide the processed data set, the pruned data set downstream, which will be a smaller data set that may be easier to read in if you have a very small amount of memory on your local machine. Before we calculate relatedness, now relatedness is done by calculating literally pairwise calculations for each subject. So we have one subject and two subjects. It looks across all SNPs in their genome that we've provided and identifies the similarity, essentially the correlation between each person's set of SNPs, just to see in this multidimensional genomic space how similar are two people's genetic sequences at the genotype locations. It takes a little while to run on 5.6 million variants. And as we saw earlier, due to linkage disequilibrium, there are mostly blocks of SNPs across the genome. The vast majority of sites of variation are actually part of these linkage disequilibrium LD blocks. They are correlated with something else, either very, very strongly or moderately strongly or not so strongly. It's not just a bunch of totally independent, uncorrelated SNPs. So we can ease calculations, and in fact, we won't get any extra information by including SNPs that are in LD with other SNPs when we're looking to evaluate the similarity between two people, because if two people have the same genotype in one location, they will also have the same genotype at all the other locations that are in LD with that. So we can generate a second pruned data set to remove a large number of SNPs, remove any SNPs that are actually in high LD with other SNPs. We will do that, I believe, in this next step for Ancestry. Actually interesting. Maybe we could do this in the opposite direction. If you're not having any issues with memory on your computer, then this will run just fine. This will read the original genotype data. It will again extract, only keep those SNP variants that passed our initial QC, It will only keep the individuals that passed our heterozygous and our missingness filter. Remember, this valid sample now only has 990 subjects in it. And then it will calculate these pairwise estimates of identical by descent, basically proportions of the genome that are identical for certain stretches and likely came from a common ancestor, quite recent common ancestor. The larger the proportion of this sharing, the greater amount of shared genetic material you have. It's quite intuitive. A relative cutoff level is basically the proportion of shared genetic information that we are willing to accept. If you had a value, the value is calculated as something called pi hat. If this value was 1, then you would be looking at a twin, a monozygotic twin, or a sample duplicate. And as you tend towards 0, you have someone who is sharing very, very little of your genetic architecture in terms of identical by descent. If we run this, oops. So I'll check this. So we're in terminal. I thought I had checked this. If I click run current chunk for this bash, yeah, you see it's not going to work because it sticks it in terminal, I believe. Just kind of disappeared. Nothing happened. Or it says it's running, but I can't see it running. So I've always copied and pasted. That is when I'm using RStudio. Yeah, got the stop button, thanks. It still runs, Jamie said. So it still runs in the background, I imagine. Is that the case? Puts the results afterward. Okay. So that's good to know. So you can, right, so you can still use the current chunk function in the R markdown to run the entire thing. I suppose it'll still write, you'll always be able to see this output in the log file that it writes, which will always update every time you run a new set of commands. It will print everything, so you'll be able to go back and check, but you won't be able to see the progress of the job as it's writing these things out. So what you'll see here is now it has to identify among all 990 subjects the relationships between people, between every pair of subjects across. Now we have our 5.6 million genetic variants. So if you run this code, it should work. It doesn't take too much memory, but it does take processing time. And with Crowdcast open and with other things kind of running, typically when I ran this code inside of the Docker with no, well, not nothing running, but no Crowdcast and no other kind of streaming happening, it takes about 2 minutes, not longer than 2 minutes, 3 minutes to go through all 5.5 million variants. So you can wait for this to complete if you'd like, but it would take a decent amount of time. We're only at 140,000 markers. It's definitely maxing out in terms of the kind of core CPU abilities here. I'll take a look at a couple of questions. I think there was a new one. What contributes to samples needing to get removed due to heterozygosity? So I think I mentioned it a bit, but it definitely bears repeating. The heterozygous markers typically, right, primarily excess heterozygosity would indicate a contamination, a sample contamination of the DNA before your experiment. So Jamie started 5 minutes ago and is at 3.7 mil. Okay, well, chugging along. I'll probably end up killing this and going a long way around. The best part is that I have it in the notebook here. Yeah, heterozygosity is true. The other one is inbreeding, which is kind of less of an issue, but it's also something that you'd want to flag, only because in those cases you're dealing with an individual that has a genetic component. Their chromosomal arrangement is a bias. It violates some of the independent assortment assumptions that we kind of take for granted when looking at genetics at the population level and not incorporating family data. So we don't want individuals who have excess heterozygosity due to consanguineous relationships, and we definitely don't want to analyze a sample that's been contaminated with someone else's DNA and therefore just has more rare alleles than you might expect them to have, and therefore a bolstered amount of heterozygosity. So Sunny's moving through 4.7 million, great, it goes. It's kind of satisfying, it's fun to see it going through all this stuff. Very welcome, Srijoy. I am going to kill it because I think we can make some more progress here. I'll use the smaller data set to perform this calculation. The reason why it's also not essential, nice to run the code, nice to see how it works. It's going to be disappointing because there are no cryptically related subjects in this sample at this threshold. It's very important to check, but this QC file that it spits out in the end will not actually have anybody in it. It's going to be the same FAM file as we have before, which is good. It's, again, good news. It's what we want. If you end up with a large number of people missing, especially if you're actually working with a smaller data set, it can be quite tragic because there's not too much that you can do about that. It kind of is what it is. So it's always good to it's always good to check. It's always very important. Now, the other thing is, and as you start to get a little more comfortable with Plink syntax and how it works, the reason why this is taking so long is because I haven't done the one step that I mentioned is possible and sometimes a good idea before running this type of analysis. It won't bias the analysis to not remove SNPs that are in high LD, but certainly for computational time, it will vastly improve the performance to use a smaller data set than the full original one and our SNP list that includes 5.5 million SNPs. So the next step, which again I mentioned I tried to emphasize at the end of the morning sessions, is the idea of ancestry. Right, we saw those principal components plots a bit so that we could identify discrete groups of people that belong to specific ancestral groups. And outlying ancestry is a really important thing to consider, especially for polygenic analyses, because as we saw, the way that the polygenic score is calculated relies heavily on broad patterns of allele frequencies, especially when you're including a lot of SNPs in the calculation. For this particular workshop, a full ancestry or admixture analysis is really not feasible. I mean, we've seen just getting some of these steps done can be quite challenging unless you have a full environment set up, ready to go, and you have experience with it. Also, performing a full ancestry analysis, for me to tell you, based on your genotype data, what proportions of which ancestries each one of your chromosomes has on it, to perform a proper admixture analysis, you need to perform a lot of fairly heavy computation using very large reference data sets. We would have needed to download dozens and dozens of gigabytes of reference files. We would not have been able to process the data on a local laptop for sure, and the time it would take to run the analysis would be far, far too much. So it can be very challenging to do that. One thing that we can do is the kind of precursor to a full admixture analysis, which is something that allows us to just identify some or any outlying observations. And for this one, we'll do the first step that I kind of suggest before, can be very helpful before evaluating relatedness. These are linked. These concepts are very linked, the relatedness and ancestry. Ultimately, the ancestry is using a principal component analysis to identify similarities at the genome-wide scale among groups of people, which is very similar to cryptic relatedness, except that the cryptic relatedness is being done by pairwise measurements of very specific calculations on allele frequencies. The first step here, therefore, is just the removal of SNPs that are in linkage disequilibrium. And the way that this works is it is a sliding window operation where we begin at chromosome 1, we go to chromosome 2, chromosome 3, and we start with a window. These parameters can be changed. They can actually have quite dramatic effects on the final amount of data that you include in your analysis. We remember some of those plots. I showed a plot in my linkage disequilibrium talk that looked like these big red triangles, and you could see the blocks that we would call those haplotype blocks, where you may have many SNPs, just a handful, a dozen, maybe several dozen, that are all in very strong linkage. What this command is doing is taking a window of 50 SNPs, and it's looking across 50 SNPs in our genetic data file. It's looking at all 50 of them, and it's taking the first SNP in that window and saying, okay, of the rest, of the 49 other SNPs, how many of them is this SNP correlated with, and what is the strength of that correlation? And if the strength of that correlation is anything above 0.2, it will start to remove those SNPs that it is associated with. So it will keep the first SNP and remove other SNPs that are correlated at that level. Then the sliding window will move 5 SNPs down the chromosome, 5 SNPs over, and take the first SNP that it finds there, right, of the remaining SNPs. And then look at another 50 SNP window. Which SNPs is this associated with? And are any of them associated above a correlation of 0.2, an R-squared value? And it will continue all the way down like a zipper through all the chromosomes, and eventually you end up just removing a whole whack of SNPs. Anything that is correlated with what was started with as the first SNP, anything that was correlated with another SNP at any high level above 0.2, anything with kind of a small, moderate, and certainly large correlation is no longer going to be in the data set. And it's going to spit out another list of variants that pass quality control. This one should run fairly quickly. We'll run this. Of course, I shouldn't say this one should run fairly quickly, because that's the #1 scientific way to ensure that it runs extremely slowly. But we have some of these same parameters in. Remember, we're keeping our quality control. So Daniel, the 5 is the slide, the window slide, taking the window size of 50 and taking a step of 5 SNPs and then recalculating correlations within that 50 window. So the step size is the middle parameter for in-depth pairwise. And the reason why you would change these parameters, the reason why step size, R-squared, and the window size are quite important to consider, and you may use different parameters in different situations, is because the patterns of linkage, so you can see now it's going through all of the chromosomes, it's zipping along, and it's removing quite a lot of variants from these chromosomes, leaving behind only a couple dozen thousand, as opposed to hundreds of thousands. If you have a very large window size, what it means is that we're going to be excluding genetic variants that may be quite physically far away on chromosomes, but actually have some long-distance linkage structure. That does occur. There can be quite complex linkage structure in that sense. By changing the window size to be quite small, we are actually preventing the removal of SNPs that are in very long-range LD. Sometimes that can be desirable. The window size, honestly, I don't know what a good reason is to change window size other than maybe computational capacity. Obviously, if your window size is larger than your or your step size is larger than your window size, then you have the potential to miss variants and miss patterns of correlation. And then just the stringency of that correlation you're removing is the R-squared threshold. And in this case, we're actually being quite liberal. We're removing quite a lot. Part of the reason for that is when we are visualizing, when we're performing the principal components analysis and visualizing relationships among subjects, you do not need an extremely high dimensional space. You do not need 5 million genetic variants in order to perform that. We want the most independent signals, and those independent signals to be matched among all the subjects that we're looking at, just the most independent ones. We are not, in this case, downstream using this data set to perform a GWAS or to perform another association analysis, in which case we do want to keep in a certain number of SNPs. In fact, we may want to keep all of the SNPs that are in high LD, because unless that LD is absolutely perfect, it may change the nature of the association of that SNP with their outcome. And it's also very useful for fine mapping. We want to keep in all of these SNPs. We can start to harness LD structure. That's right, holy moly. It removed most of the variants. So it gives you an idea of when we have a very liberal threshold, 0.2, we are actually going to end up removing quite a lot of genetic variants. And this is good because in the next step, what we're going to do is write out a new whole data set. We're going to write out a new BIM, BED, and FAM file. So in this case, we will take our original genotype data. This time, we're going to extract the output from that code we just ran, the indet pairwise, which creates a file called prune in. It's telling us to keep this set of genetic variants because these do not violate the pairwise LD calculation that we specified here. We'll also keep our valid samples. It removes those 10 that have high levels of heterozygosity. And then this command make bed will actually now, instead of just giving us those quality control files like lists of SNPs or lists of subjects, it's actually going to regenerate the entire BIM, BED, and FAM trio of files. And all three of those files will have this Geno4PCA prefix. And then what we can do is use Plink again to actually take that much smaller genetic file, which now, once this is calculated, we can provide this on the Fig share. I can upload it into a separate directory if you want to try operating on it. Because it has so many fewer SNPs, the file itself will take up much less RAM to read in. However, it's also going to create some problems when we want to calculate our polygenic risk score, and I'll explain that when we get to that stage. So it says writing. I suppose it's taking its time. It's really quite remarkable, actually, how much the process is slowed by performing this live broadcast. I should get my resource manager out and see the types of demands on the CPU from this Docker versus the Crowdcast. So I guess you never know until you know. We did perform pretty rigorous testing. So we had several people test these codes to make sure they'd kind of work. But I apologize for excess blank time. I think it leaves us a nice opportunity to discuss any questions or address any other concerns that people have, including things that might be related to other topics that I presented this morning, or any general curiosities that you might have. Because this should finish writing soon and we can get to it. And we will get to the precise calculation, which is actually coming up as soon as we've taken a look at this principal components plot. We'll generate one more file set and then get right into polygenic risk score calculation. So if there are any questions, here we go. Somebody's asked a question. True or false? Most of Plink is QC. Well, I'm wondering, do any of the students have thoughts on that? Any votes? Interesting. So true or false? Plink is mostly QC. The answer is 100% true. Most of any analysis of this kind is QC. Most of bioinformatics is QC. Pulling data sets. Brian says, don't think so. Oh, is this in relation to Daniel's question about optogenetics? So in terms of my understanding, optogenetics is not applied in humans. I think there are experiments in non-human primates, but if Elon Musk has anything to say about it, then he's one step away with his what's called a neuronet. The neuronet, which is purely electrical, but I'm sure the problem with optogenetics is you need a genetically modified organism. You need a substrate. You need the rhodopsin reporter. So without germline genetic modification, I think we're looking at into quite deep dystopian future when optogenetics becomes practical for humans. It's when humans become the experimental model. That's when optogenetics is. But yeah, Elon Musk is. I'm sure he dreams about it. So here we. Nothing against Elon Musk. I just realized this is recorded for. This is recorded for posterity. So yeah, Elon Musk is great. NeuroNet's a little scary. I'm sure you could convince me otherwise. So here I'm going to run this one. And I know some of you are likely kind of moving along in terms of this. Hopefully it just doesn't take long for it to go. This is simply just taking that list of SNPs that we left in after pruning for LD, and it's going to write out a new data set, a smaller data set with just those SNPs that passed QC. And so hopefully that doesn't take too long. We'll do the PCA. We can read out the PCA tool. So here we go. It's a bit much faster now that we only have 300,000 remaining independent variants, extremely independent variants. Although it's still taking its sweet time to write the file, because these read-write operations are kind of under high demand when I wonder if it actually does improve. I mean, the download improved when I turned off my video. I wonder if the write speed for this improves. It does look like it has improved some of the speed. Now everyone can't see my face. Feels even more removed. In vivo gene editing in humans. In muscle cells. Humans. Jeez, I'm not aware of any reputable published literature on active gene editing in humans for therapeutics. I've seen it in pigs. This is like very George Church. I look at a lot of George Church's kind of latest editing. Oh yeah, retina. Interesting. Let's see if we're missing any questions here. Regarding the removal of SNPs in LD, how does it decide which is the SNP that stays and the SNP that's deleted because they are now LD, p-values, betas? Great question. So there's a distinction to be made between two procedures. One is LD pruning and one is clumping. What we've just performed here in Plink is LD pruning, which is not informed by any type of association statistic. It's not informed by any functional effect of a SNP, because Plink is blind to GWAS data. We haven't given it GWAS summary statistics. We haven't told it which SNPs are actually important relative to each other. LD pruning is a blind procedure. It starts at the beginning of each window. So we have a 50 SNP window. It takes the first SNP in that window and checks for association, correlation, linkage with all of the other SNPs in the window, and then shifts that window by the step size. It is completely agnostic to the role that those SNPs might play, whether they might appear in summary stats for a GWAS or whether they might not appear, whether they were imputed or genotyped. It has no idea. This is problematic. This is potentially problematic. It's one of the reasons why it's okay, it's fine to use the method when we are looking at things like ancestry and cryptic relatedness, but it's why it can present problems when we use a pruned data set and try to match it with GWAS summary statistics. Because the GWAS summary statistics often, I mean, if we have the correct stats, the genome-wide marginal effects for all SNPs that they tested, we may not be able to match. We may not actually find the SNPs that are the most important SNPs in the GWAS in our pruned data set because it's pruned blindly. Clumping, by comparison, works in a very similar way, but it does use the p-values and the beta coefficients. I think it's pretty much just the p-values in most clumping algorithms. PRSice does this for us, which is why the run that I have and in the best practice for what you'd want to do when matching our genotype data to GWAS summary stats and then performing a polygenic risk score analysis, we start with un-LD pruned data. And then once we've matched the top SNPs, basically all of the SNPs that we want to include in our score for a given threshold, and we find them in our data set, then we start with those and identify SNPs that are correlated with them and remove those signals. That is clumping, because essentially we're removing the LD structure around specific clumps of significant variants according to our analysis, rather than a blind pruning. So I hope that answers the question. And there was one more question. I mentioned in Alzheimer's GWAS, most of the risk variants identified in Alzheimer's reside in non-coding regions of the genome. That's true for most complex traits. Is this the case for major depression? Yes, I believe it is the case for major depression. You mentioned that very few variants have been identified and reproduced. So, right, until recently, very few regions and variants. Right now, there are over 102 independent variants that were identified in the meta-analysis, which is, kind of on par. I mean, it's actually better than Alzheimer's disease. Alzheimer's disease has less than 40, depending on who you ask, independent regions that are associated with Alzheimer's. Schizophrenia has, I think, now 250, most recent analysis. So major depression is actually doing kind of okay right now in terms of the number of variants that are being identified. And this is, I mean, it's a great question that kind of segues into tomorrow. Identifying the downstream function of non-coding regions of the genome has really become the focus in the post-GWAS era of psychiatric genomics. As our understanding of the complex function of the human genome evolves, we can start to understand what possible influence a non-coding change in, what even, 10, 15 years ago would have been called, quote, junk DNA, that people thought was just a graveyard for old genes that have, kind of become evolutionarily obsolete, for copies, aberrant copies of viral DNA that have worked their way into the human genome, for jumping genes and transposable elements, basically just parts of the genome that have nothing to do with the structure or function of organisms. Now it's understood that most of the function, as you point out, most of the important disease-related genetic variation that we observe, especially from GWAS, is actually in these non-coding junk regions of DNA. But we are learning more and more. I mean, every year the number of ways that we can annotate and validate specific points of genetic variation for potential functional consequences grows enormously. So it's really important to consider how non-coding DNA can actually interact with regions of the genome to influence things like the expression of genes, so-called expression quantitative trait loci, both in cis and trans, nearby each other or distantly on other chromosomes because of the 3D conformational structure. They interact with the epigenome in regions that do not actually affect the structure of proteins after translation, but they can affect the way that methyl groups attach themselves to CPG islands near genes or around genes. They can adjust the way that histone tails are modified with different types of acetylation or methylation, and we can also look at the way that non-coding DNA opens and closes chromatin according to other epigenomic alterations using different types of sequencing, such as ATAC-seq, to figure out why there are certain points that on the genome that look like they're isolated, but actually show very strong associations with different types of molecular and clinical phenotypes. It's a whole ongoing investigation. Tomorrow we'll discuss a bit more about that with Srijoy and Leon. So this analysis has now completed. We have slowly come to the point where we've written out a relatively clean, we've pruned for a number of different quality controlled measures, including removing SNPs and high LD. We have 300,000 variants remaining, and now we can perform a principal components analysis on those 300,000 variants to try to identify if there are any individuals in our sample who may not actually be like the others in terms of ancestral background. And remember from that example from Invar in the morning, if you calculate polygenic scores in individuals from diverse ancestral backgrounds, the distribution of those scores will not be normal across those populations. It can greatly bias association results downstream. So it's very important to make sure that we have a relatively homogeneous ancestral population that we're working with. And that's what we're currently doing right now. The principal components analysis is looking across 300,000 variables and collapsing them. Well, not really collapsing them, it's transforming them into latent variables that maximally explain variation in all of the input genotypes. We are getting these eigenvectors, which we can use R to read in this output file that it's generated, eigenvec and eigenval, these decomposition matrices from our original input data, which provide us with actual components, these latent variables that explain the most amount of variation in our genetic data. And if we run this chunk of our code, you can see we're just reading in the eigenvec file, which is all of our principal components. So each individual will have a value for principal component 1, for principal component 2, for principal component 3, and simply plotting it in ggplot gives us this plot. Principal component 1 is on the y-axis, principal component 2 is on the x-axis. These are the latent variables that explain most of the variation, linear variation, in all of the genotype data among these subjects. And you can see that most individuals are clustered fairly well together. Some individuals may actually have some admixture. They may have components of their genome that are not belonging to the same ancestral line. It's very difficult in this case to make any concrete conclusion about it because, as I mentioned before, one of the limitations of doing this in the context of a workshop like this is that usually we would need to run this principal component analysis with our genotype data merged with a very large reference panel of confirmed ancestral backgrounds. And that allows us to see the scale of these components, which are scaleless. We cannot interpret this distance as being meaningful in terms of genetic diversity. However, if we run the principal component analysis of our subjects alongside including data from another 2,000 individuals where we have very solid ancestral knowledge from, we can very quickly ascertain if this range of variation is within a Caucasian European background. All subjects are self-report Caucasian. Do all of these individuals fall within that group? Or is it actually the case that some people down here overlap much more strongly with individuals of East Asian or African ancestry? So this is a demonstration of principle. This is a demonstration of something that you can do. It is computationally expensive, and also it's expensive on memory and storage to run a proper admixture analysis. So if you want to, you don't have homogeneous data as representative of the general population, right? So it's an absolutely vital step when looking at large populations that do have admixed groups or diverse groups and underrepresented population. And typically the step that would be taken in an analysis like this, if, for example, we found these individuals toward the bottom to actually be of African American ethnicity or African descent, and everyone else fell on a spectrum of Caucasian or European ancestry that we found to be within acceptable parameters, then this group we would actually exclude entirely, only because statistically we have so few subjects that performing downstream analyses on this group would be too little. Now, in other groups where we have tens of thousands or even hundreds of thousands of subjects, we would perform stratification, population stratification, where we'd split the sample and then perform separate analyses inside of each group separately. That's the idea behind that. Do you also use T-Sne for finding genetic clusters? So T-Sne is also like a kind of multidimensional scaling approach. PC is more common for determining broad ancestry. There are, I think, some reasons for that have to do with the structure of genetic data as opposed to gene expression data, which is typically where you see T-Sne applied more frequently. I'm expecting to see some of that discussed tomorrow. I imagine there will be some of that possibly. They're getting at very similar ideas, but PCA is usually preferred for identifying broad scale and ancestry. I wish I had a better answer for you on why we don't use T-Sne so much in the literature, but it would be interesting to look into. Okay, so it's similar conceptually, but the way that the principal components analysis is done versus the compression in T-Sne and the dimensionality reduction, apparently this is a more optimal way of doing it, or preferred. Perhaps it's historical. It would be a great homework question. We'll bring you up on stage tomorrow, and you can tell us I imagine it has to do with the quality of genotype data as opposed to the other, but maybe tomorrow you can come on stage and share your findings. So we've made it this far. We've made it quite far. The one thing that's important to know is in generating this principal components plot and generating the eigenvector table that contains the principal components, we're also creating essentially a new phenotype file, because in the event that we do include everybody in this analysis, which we will, we may want to covary for these principal components, perhaps the top 5 principal components. We may want to try to remove linear variation in our phenotype data, and we want to control the analysis between a polygenic score and an outcome phenotype for individual differences in kind of population microstructure. We would call it microstructure when individuals are kind of still within a cluster of a single ancestral background, but still have some differences broadly across the genome that we see in this distribution across PC2 and across PC1. So we can covary for these distributions in downstream analyses, and that is indeed best practice when we're doing these types of analyses. In fact, we do this as well when we're running a GWAS. We calculate this principal components analysis before looking at individual SNP associations to try to account for any broad scale variations in allele frequency due to ancestry as we calculate the association of each SNP with our outcome. The final generation of the clean data set. This is where I was hoping we'd be a little earlier, but I think it's great that we've made it to this point. It's not fully necessary at this stage. The reason why I say that is because, as I mentioned before, due to differences in LD pruning and the clumping procedure and the fact that PRSice performs its own clumping when we calculate polygenic scores, we want to stick with the full large data set. We want to be able to match the maximum number of significant SNPs from our GWAS to our available genetic data, but the memory concerns for any person here who's having issues reading in the full data set, just for demonstration, we could just stick to this data set that we used for PCA, the Geno for PCA, which had this LD high LD SNPs removed from it. So this will be a smaller data set, and I'm going to replace in my code this Geno clean data set, which would be bigger, but is again just pruned from our initial data set. For our calculation of the PRS, I am going to use the smaller data set to save some time, the Geno for PCA data set. So that will be the change. And if you've successfully run these codes, the generation of the Geno for PCA file here, then you'll be able to do the same as I go to my final set of code. So yes, Plink is dominated by pipelines for data management, conversion, and quality control. It turns out that those are by far the most important parts of dealing with genetic data sets. Analyses can be very quick, especially as new tools come around. Even the precise calculation can be done very quickly, depending on how many live streams you're doing and what else is going on your computer. But we will see. The next key part, once the genetic data is taken care of, the summary statistics. And I will just do a head of these summary statistics here. So yeah, the chunk did work. So these are the summary statistics that are taken from the Psychiatric Genomics Consortium, an analysis of UK Biobank individuals, and it's the major depression outcome, and it's a full genome-wide summary statistics. It's quite a large file. It's a few hundred megabytes. But it has all of the essential information. The marker name, the alleles, A1 and A2. A1 is the effect allele. It comes with a README file. It's always very important when you're doing these types of analysis to read the documentation, read the README file that comes with it so that we're not mistaking A1 for A2 or vice versa. It gives us some additional information. Freak, what is the allele frequency for A1 in the sample that they actually analyzed? So that's extra information. We don't actually need that to calculate our polygenic risk score. Here's the log odds ratio. That's good. That's an effect size. Here's the standard error of the log odds ratio. And here's a p-value. So this is a fine-looking summary stats file that PRSice will be very comfortable reading. All we have to do is tell it where each piece of information is stored. Which column is the effect allele, which column is the SNP ID, which column is our effect, which column is our p-value. And also, what is the statistic? What is the type of effect? Is it a beta coefficient or is it an odds ratio? Right? This has to do with whether or not your outcome was binary or continuous. Did you use logistic regression or linear regression? One thing that we do need to do, unfortunately, is if the effect for a binary outcome is given to us in the log scale, which is quite often the case, we may need to transform it back to a proper odds ratio. Taking the log of the odds ratio gives us a negative positive scale. So we can kind of gauge if a value above 0 is a positive risk and below 0 is a negative risk, whereas the odds ratio, again, remember, is centered at 1. The ratio of 1 is an even balance between risk and protection. So we need to put this log odds ratio, back into the odds ratio space, and that's just done with an exponent. And we can actually do that in bash, and it's much more effective to do it in bash using a combination of awk, which is a kind of a text reader and a text editor that operates in bash, and sed, which is also an inline editor. Essentially, what these two commands are going to do is read our summary statistics file, calculate the exponent of the fifth column, which is our log odds ratio, and then generate a second file called the odds ratio with the header OR. And the second command is a paste command, which simply pastes this new odds ratio file in with the original summary stats and creates a final compiled summary stats file called MDD sumstats, which we use for our final analysis, because now we will have a column with odds ratios in the correct scale of odds ratio rather than log odds. And that's really all we have to do to these summary statistics. We won't always be so lucky. Sometimes things will be very messy. Sometimes SNP names will not actually be RSIDs. Sometimes the SNP names will be presented as chromosome, location. If that's the case, then you have to be extremely careful when matching summary statistic data to your genetic data set. You either have to go through a series of steps to annotate all of your SNPs based on chromosome and chromosome location into RSIDs, which requires, depending on the size of your data set, a couple of different tools to manage that. Or you have to, you may also have to lift over. There's another idea, as I mentioned briefly earlier, as we constantly update our knowledge of the human genome and our physical map of the human genome, the position of SNPs may change according to the latest reference. So there are different genome builds. They go under the title human genome 18, human genome 19, GRCH. What does that stand for again? Genomic range, chromosomal 38. There there are these updated versions of the human genome every few years. And every time that happens, new tools are developed, and the latest results that come out, people try to update it so it's in the latest format. This means that if I'm using a genetic data set that was imputed and quality controlled back in 2017 or earlier, 2015, 2014, and I'm using summary statistics from a brand new GWAS that's using the latest build of the human genome, if those SNPs are presented to me as chromosome and location, those chromosome locations will not match. You will have to perform a liftover procedure through a genome browser. Again, as I said earlier, it is a messy, messy, dirty world dealing with these types of data, dealing with the tools and the versions of the tools. It can be very unpleasant, but it can be very rewarding. If you can get it to work, if you can recognize the key pitfalls and the opportunities where things can go awry, then you'll be much better off when you know what you're kind of aiming to look for. So each of these steps are kind of essential to arriving at that final moment of success. I see, for some reason because I'm in console. Well, that's what you get for. So now I'm running this opt command to simply transform the odds ratio. You may be thinking, I mean, if you wanted to do this in R, you could, but then you have to read a 350 megabyte text file into R, perform the calculation, and then write it back out again. It's actually much faster, more efficient to do it in Bash, right from the command line, operating on the text files themselves. So we have a couple more questions I can get to right before we run PRSice and declare success. Triplet studies. Sunny asking triplet studies where 2 parents and a child are included as units. How often is this done in GWAS studies? If not always or never, does it change how we handle ancestry and relatedness? So these triplet studies are not usually part of a GWAS framework, although you can incorporate GWAS data from family data, or you can perform a GWAS type analysis in families. I have virtually no experience doing family-based association testing. One thing that in terms of relatedness, obviously it becomes a benefit. It becomes a benefit in terms of how we understand causal relationships between variants. If you have genotypes of both parents and you have genotypes of a progeny, then you know for sure that the allele, the actual state of the allele or haplotype that you observe in the progeny came from one of the parents. You know exactly where that came from. You know that that allele is identical by state, and the statistics account for that in certain ways. When we don't have family data, we're making assumptions about how people are related to each other and how different SNPs and haplotypes affect our causal outcome or affect our outcome in a potentially causal way. With family data, we have a much better handle of what may be causal and what may not be causal. In terms of ancestry, though, family data doesn't really change our understanding of ancestry. We're still requiring a large annotated reference panel for each person in that study. In fact, it is further complicated by the fact that in that one generation, you may have admixture occurring, depending on the ancestry of both parents. And I mean, having never done a kind of genome-wide family study at a large scale, I honestly don't know how that type of analysis would be handled. It seems complicated enough to me without the family data, but I'm sure that it offers a lot of very interesting benefits when handled correctly. So basically, neurobioinformatics is simply like science in general. Most times it doesn't work. When it works, we dance of happiness. Do we dance of happiness? Do we dance of happiness when it works? Yes. Yeah, I've seen it. I have seen the dance of happiness when it works. And it is like science. As a very talented and lovely cognitive neuroscientist Colin Hocko once said, and since has said several hundred times, science is hard. Neuroscience is hard. And he's right about that. So we should take our victories and celebrate them and acknowledge them. And you're absolutely right. Most of the time, it doesn't work. But it doesn't work for a while, and then it works. There are no people who are flawed because they can't get it to work. It's just a matter of time until it does. Hopefully, we have provided kind of enough for everyone to mixed feelings. That's my sort of question. No, it's the goal is to get it to a point where it works most of the time. I think that should be the goal. You get it to the point where it works most of the time, and then you feel more able to cope with all of those times that it doesn't work. And I think that everyone's capable of getting there. So we finally made it. We've made it to the grand moment. We've made it to the point that I said we would get to. We will calculate polygenic scores for these people. And we will do it using kind of state-of-the-art PRSice version 2 software at a high-resolution level using high-quality genotype data. Most of that is true, and we have arrived. Most of these points talking about how we consider our calculation are relevant to what I've kind of mentioned during the slides in the morning. Questions that everyone should consider when thinking about calculating a PRS. What p-value thresholds do we want to include, right? Do we want to try to calculate one polygenic score that incorporates some optimal number of SNPs? How might we do that? Do we want to set a number of a priori thresholds like 5 times 10 to the negative 8, that arbitrary genome-wide significant finding? What if our GWAS had no significant GWAS hits? What if there were no significant SNPs at the genome-wide level? Do we even want to calculate a polygenic risk score there? How do we want to deal with the issue of LD? Are we going to perform clumping, or are we going to prune the data set before we merge? These are things to consider. Which type of PRS calculation do we want to make? Now, I've only gone over one, which is an additive, a weighted additive, and divided by number of alleles, kind of standardized, typical additive PRS. There are other formulations for polygenic risk scores that can offer different advantages and disadvantages. Which allelic model do we want to use? That's another question. So I've talked about allelic dosage, and we've talked about genotype data. Each SNP, each biallelic SNP, you can have one of 3 genotypes, right? That can also be coded as a 0, 1, or 2 if you reference one specific allele. But there are other models of genetic effects in terms of which combination of alleles we might expect to actually be producing a change in a phenotype. For example, an additive effect is one where every time you increase the number of C alleles, for example, or A alleles, you have an increase in the phenotype it is associated with. So that is additive because every time we add one allele, the value of the phenotype changes linearly. In a dominant model, you actually need one copy of a dominant allele in order to see an effect. You only need to be heterozygous for the allele that's having the effect in order for that effect to happen. And then, in fact, it doesn't actually matter if you have 2 copies of that allele. It is one dominant effect. It's only when you do not have any of those alleles that you see a deficiency or a difference in that. Recessive is the opposite, the inverse. It's actually alongside dominant if you flip the IDs of those alleles. If you now change your reference allele, a recessive effect is a difference in a phenotype that's only observed if you have 2 copies, usually of the minor alleles. A recessive effect or recessive phenotype would be one in which you need 2 of the minor alleles, the homozygous genotype, in order to see that phenotype. And you can change these. Really, all this does is change the numerical coding of the genetic data behind the scenes before you calculate the alleles. So instead of calculating 0, 1, or 2 for the number of alleles, maybe you only count a 0 or a 1, right? If it's dominant, we only care if you have 1 copy. So there will be no instances where you have 2. Perhaps recessive is the mode of inheritance. So maybe we only care if you have 2 of the minor alleles. So it's all about coding behind the scenes. There are a lot of steps that are done automatically behind the scene in PRSice. One of the things that we need to consider is, where do we draw the line in terms of calculating our polygenic score? And Daniel's asked this question about calibration, which is a great point. The idea is, how do we know that we've included the right number of SNPs, or what is the optimal number of SNPs to include? There are different ways of doing this. Some of them are based on penalized regression methods. Some of them are based in what we're going to do here, which is a high-resolution p-value threshold testing, basically introducing more and more and more SNPs, calculating hundreds or thousands of polygenic risk scores, and examining at which point we observe the optimal association, the greatest amount of variance explained, of our PRS score with our phenotype. And that will always be an overfit estimate. So it's a first step. It's always going to be an optimal association within the sample that we are testing. And there are steps to follow after that we can discuss in later days, perhaps even on Friday, when we'll bring some of these numbers back. The other two steps, which are kind of pains, but we don't have to do because PRSice does it, check for ambiguous alleles. So an ambiguous allele would be a SNP that is a polymorphism where one allele is A and one allele is T. And in that case, you may remember, A only can bind to T and T binds to A. So if you have the A version of the allele, then your rung will be A on top and T on the bottom. If you have the T, then T will be on top and A will be on the bottom. But unless you are absolutely sure from every single stage of your analysis, from the very beginning of your genotyping through to processing, possibly leftovers to other genome builds, the imputation pipeline, the quality control, unless you are absolutely certain that you have oriented the genetic data consistently and perfectly so that your top and bottom strands are aligned, then you cannot be 100% certain whether or not you have a T or an A, right? Because remember, we only need one side of the DNA double helix to infer what the other side is. If it's a T, the other side's going to be an A. So unless you're keeping track of strand orientation, and strand can be a really big pain in the bum, you might not want to include those ambiguous SNPs in your calculation because you could throw things off. Again, just in case your strand assignment hasn't been maintained perfectly. And then finally, allele flipping. And that is just that fact. If strand has been dealt with differently between your GWAS summary stats or between leftovers and different processing pipelines leading up to your genotyping and leading up to the processing of your genotype files, then if you have a G to T SNP, somebody may have a C allele. Well, they don't really have a C allele at that strand location, but C binds G, so we know they have a G. So in this case, we can just flip alleles and match the genotype data accordingly. Or it's also possible that you have the misassignment of a major or minor allele, and that will be something that happens quite frequently. You can have it backwards. You can actually have differences in allele frequency, especially for alleles that are quite common, that are close to 50%. Then the minor allele and major allele may flip in the population. Now, if you have a lot of alleles and a lot of SNPs that have low minor allele frequency that are flipping between major and minor allele, the one that is more common and not common, then you may have a major problem. So what we're going to do is we're going to change one of the things about this line of code, and that is just the input data set. Remember, we're going to change, again, this is just depending on your computational needs and mostly for timing for us. I'm going to change the target data set to be Geno for PCA. You see each one of these options is, I've detailed some of them, and the PRSice website that I linked above also describes a very large, it describes all of these options in details. But in short, we have our base, which is the summary statistics that we've added odds ratio to. We are informing the program that we want to calculate, that we are going to be looking for an odds ratio, that our summary statistic is in fact an odds ratio and not a beta coefficient. We're going to tell it the p-value column is p, the SNP ID column is marker name, and our statistic column is OR. So remember, we calculated the exponent of the log OR, and now we have that. Now I also have a separate file of phenotype data. And this is just a text file, but it has all of our subject names, just like in the FAM file, correspond to the individuals in our genotype data set. And we have a large number of variables in here. I've also merged in the principal components from when we ran PCA, that eigenvec file. I can tell PRSice now to calculate a whole bunch of polygenic risk scores in our data and run an association analysis of all those polygenic risk scores against a variable, in this case, a score of depression, depression 5, a five-item list of depression. The binary target is now telling it that this depression variable is not a binary variable, so we're not going to use logistic regression, we're going to use linear regression for our analysis. And I can also specify covariates from the same phenotype file. I'm going to include biological sex, I'm going to include the subject's age, I'm going to include educational level, these potential confounders of depression, and then I'm going to include the top 5 principal components that we calculated from our genotype data to help adjust for smaller amounts of population microstructure. And then finally, I'm specifying the target genotype data. This is where we'll be calculating our polygenic risk scores in, and that's what we've just generated, that smaller pruned data set. And these are the parameters that tell it how many scores to calculate. Every time we give it a p-value threshold, it finds the SNP that is right below that threshold, right? A SNP that has just a smaller p-value than that, and then finds all of the other SNPs that are at a p-value threshold that is even below. So it's cumulative. It's going to start at a lower range of 5e to the negative 8, which is that genome-wide significant association. So the very first polygenic score it calculates, just as I explained it would calculate in the morning sessions, it will take all of the SNPs that are more significant than 5 times 10 to the negative 8. And then at an interval of actually 1 to the negative 5, it will increase the p-value threshold and calculate another polygenic risk score, including all of those SNPs. And it will continue increasing in intervals of 1 to the negative 5 until it's reached a p-value of 0.5, which of course is not significant by any mean. But the fact that we are calculating weighted polygenic risk scores means that as we approach a higher and higher p-value, as the p-value becomes less significant, the number, the magnitude of the effect, that odds ratio, is also getting closer and closer to 1. No difference in the odds of the disease from the GWAS summary stats as we increase. So despite the fact that we're now going to be including tens or hundreds of thousands of SNPs, or maybe even every SNP in the entire genome in our analysis, which I think it will do by default, it will also calculate one polygenic score of every single SNP in the summary stats. The majority of contributions of those SNPs that are toward the higher p-values will be closer and closer to 0. Now, one of the things that we have to think about when looking at different p-value thresholds is the interpretation of the association of those scores changes because we are including different regions of the genome that may have different biological functions, different involvement in different biochemical pathways. So this code here, I will run in terminal. And you see it's going to read in the summary statistics file. It should take, about a minute, sometimes less. And it's a full genome-wide summary stats file. So in your own adventures, you will encounter summary stats files that look extremely similar to this, several million variants. And it will tell us how many variants are in the SumStats file. It will tell us how many variants are in our input genotype file. And it gives us fairly useful outputs in terms of what's going on in the calculation. I guess here I've specified no full, and that is to clear the default, which I mentioned before, which PRSice will do, which is to automatically calculate one polygenic score at a p-value threshold of 1, so including every single SNP that is in the summary statistics file in that calculation. Another thing that's happening under the hood that I discussed briefly earlier is clumping. In the case of this particular run where I'm using the pruned PCA file, it's not really important. Pruning the clumping won't really make a difference because we've already removed a lot of the LD structure from our data set. But when we give it the full data set, every time it calculates a new polygenic score, it identifies the top associated SNPs and it matches them in our genotype file. And after it matches those, only after it matches those, it prioritizes them and then runs clumping to remove any signals that are dependent on each other or not independent. So it does that because now it knows the p-values and knows the significance of each SNP. It can take the top signal from each region and then remove others around it that it's associated with, rather than a blind pruning of the genetic data set, which is much preferred. So here it's read in over 8 million variants in the base file, which is our summary statistics file. And now everything's going very quickly, which is good. It's giving us information on our phenotype data. So we can scroll up and take a look at what's going on here. It's excluded 730,000 ambiguous variants by default. So remember, those are the A, T, C, G. And then it's also included 4 million variants at the end of all of this QC from the base file to match. But you can see that a very small proportion of those variants, in this case, there's 173,000 variants from our much smaller, I think 300,000 variant file, were not found in the previous data being the base file. There was one variant that had mismatch information, so that means between our imputed data and the GWAS summary stats, the alleles did not match. And we got rid of that SNP because for some reason, despite the fact that we're using the same genome build, we have the same RSIDs, something was wacko. One variant is okay, and that's great. In the end, we end up calculating our polygenic scores based on 127,000 matched variants. It's telling us which phenotype file we used, and it's telling us the results of the clumping procedure. So it performs its matching and then identify the number of variants after clumping as 64,248, the final number of independent regions matching between our quality controlled genetic data and the genome-wide association study summary statistics. We processed one phenotype. It is a continuous phenotype. There were 4 samples without phenotype information. Our final sample N is 986 subjects. All of our covariates were non-missing. There were no missing observations for each one of them. And they were all continuous or binary variables, so they did not have multi-level factors, which would have had to be treated differently using dummy variables, which they can also do automatically. And we can take a look at the summary statistics. What I'll show you is the PRS file inside the PRS directory. And if we calculate, or if we just print all of the other chunks have run perfectly for me, but this one gives me output in red text. So there's a little bit of red text at the bottom here. I don't know if that was what you might be referring to, may not be significant. It's given us a flag. It said there's one region with a p-value between 0.1 and 1 to the negative 5. This is in fact saying that one of our scores, which I'll show right here, PRSice. This is a very large file. I will head that file instead of cat. Maybe if I do this, it'll be there. So this PRSice output gives us, we only looked at one phenotype, so it hasn't listed the different phenotypes. We only had one. For every p-value threshold that we analyzed, starting with 5 to the negative 8, and then increasing at intervals of 1 to the negative 5, I believe it was, or 5 to the negative 5, or 1 to the negative 5, it has run the linear regression with covariates of the polygenic score against our phenotype, and it provides an R-square, which is a proportion of variance explained, which you can see is actually quite low, and a p-value for that association, as well as an effect coefficient, beta coefficient, and standard error. It also tells you the number of SNPs that were included in the analysis at each time. So I replaced the PCA data set into this command. So if for some reason, I switched out the PCA. I just want to make sure that everyone's doing that, because the command as it's written will have, under the target option, GenoClean. So unless you ran the final plink command here, that is line 268, that creates this final data set, it won't be able to find the target data. So I'm just using the smaller PCA data set. So this is a new set of summary statistics, but now we're not getting SNP-wise summary statistics. We're getting a whole list of high-resolution scores. And you can see each time we increase the p-value threshold to include more genetic variants, we're including more and more and more SNPs. So here, at the genome-wide significant threshold of 5 to the negative 8, we calculated our weighted polygenic score with the top 32 independent SNPs. And again, it's fewer than the 102 that are claimed in the manuscript, because we've performed quality control and that kind of agnostic pruning for this PCA data set, that we would have a much higher number of SNPs, or a much closer number of SNPs matching to if we use the real full genetic data set as opposed to the pruned data set. And I'll show, just for example, what this looks like graphically. We can, in R, just read this file in. And I will do that in our console here. I will run the chunk. And what we do is we see here all of this is actually on a log scale on the X, p-value thresholds that we assigned. And on the top is the p-value for the association of the polygenic score with our phenotype, with our depression phenotype. And so what we can see is at very low p-values, so this would have been the first, this is 5 to the negative 8. So again, this is a log 10 scale, so negative 8 is here. There is some association, a negative log 10 p-value association, somewhere around, this would be 0.05 almost. But as we included more SNPs into the calculation, the strength of association also increased. And then it kind of went down for a bit. Perhaps we weren't including the right regions, and then it shot up again right here at a p-value threshold. So there is some threshold, some point where we have maximized the association of our polygenic risk score. We've included a certain number of SNPs such that the association is the highest at any level. And that is actually given to us. That threshold, that specific threshold, is given to us in an output file called mdb best, the .best output file. Although this is the actual scores per subject. So if you wanted to end up with one polygenic risk score after going through this high resolution scoring procedure to maximize the fitting of this, then your FID and IIDs are here that match our genetic data set, and the actual value for the polygenic risk scores are here in this column. And this corresponds to that point in the full precise output where the polygenic score is of maximal association with our outcome. The code after this, which I won't bother going through because I want to be wary of time, and I think we're pretty much going to wrap up for today, is the similar operation, except performing no regression at all. In this case, we're not performing any built-in calibration. We're not interested in assessing the association of a score with a phenotype in PRSice specifically. Rather, we're providing just a set of different thresholds, just a few, and telling it to spit out the all score, using this all score function, just to spit out a file where we have all of our subject IDs and then each of the polygenic scores that are calculated at these different thresholds. And that will give us an all score file that will have our PRSs for all of our subjects at these given thresholds. You could use the same flag all score in our previous command here that is actually running the regressions, and it would spit out every single one of these polygenic risk scores into a file. And if you're doing very high resolution scoring, which we are, remember this is on a log scale, so there's quite a lot of thresholded scores that were tested between this region here. We can have it spit out all of those and just get a giant all score file. They warn against it in their documentation. So I think that's where I'm going to end it for today. The polygenic risk scores are genuine. I will update the data file, although it will be slightly larger, with the with the polygenic risk scores that will be used in tomorrow's analysis that will carry over. But the scores are genuine. The data are real. The MDD summary stats are real. And certainly if it hasn't been 100% successful in running through all of the code and getting everything calculated, you've certainly been exposed to some of the real situations, some of the real types of quirks and features that you encounter when dealing with large files, and that we encounter when dealing with kind of real world data that you won't see in these small toy data sets. So I hope it was informative. I hope it wasn't completely overwhelmingly frustrated. I think day one is always a challenge to try to get everything set up, to try to get everyone accustomed to the Docker environment. It's the first time that we've put this on together, so we really appreciate all of your patience and all of your questions. Again, everything is saved, and everything is, you can immediately look back on any of this material. So we will continue to update the data, and certainly by the end of the course, have a final release, make sure that everything is up for you and for your friends and anyone else to go back and revisit at any time. And we welcome more questions at any point, and we'll be back here tomorrow morning for day 2 with Professor Tripathy and French, who will lead our next day's festivities. So I appreciate everyone and the time and attention, and I hope you have a great evening, and we'll see you all tomorrow morning. That's right, actually. I should, Radha, thank you for reminding me. I really do want to thank the TAs for today and all of really everyone who was helping to communicate and problem solve across all of the groups. But really, thank you very much, the TAs taking their time. Sejal and Michael, I know Sunny helping out as well. And of course, Aaron, who's put the whole thing together, essentially stitching all of the pieces together, really making it work and flow. It's an incredibly challenging thing to do, given the limitations of what we have, given the strange times, a lot of big hurdles to overcome, and it's really remarkable to see how she's managed to put this together. So we'll still be in the Gather Town, I think, if you have other questions. But again, I really want to thank the TAs, Sejal and Michael, and thank you, Aaron. And for the second time, see everyone tomorrow morning.